are back with us at the 14th International Research Conference of General Sir John Kothalavala Defence University. We now move on to the fourth and last technical session for the day. This session will be chaired by Dr. Varuna Navaratne and Lieutenant Colonel Dr. Manoj Bandara. Ladies and gentlemen, I have the honor of introducing the chairpersons of this session. Dr. Varuna Navaratne is a senior lecturer of microbiology at the Paraclinical Department at General Sir John Kothalavala Defence University. He is the consultant microbiologist at the University Hospital, KDU. Lieutenant Colonel Dr. Manoj Bandara is the preclinical department head and senior lecturer in biochemistry at General Sir John Kothalavala Defence University. We have two eminent academics judging this session, whom we are grateful to have here with us today. Professor Vimaladharma Abbevikrama, Professor in Parasitology at General Sir John Kothalavala Defence University, and Dr. Jayanta Balavardhana, Senior Consultant and Head of the Oncology Department at University Hospital, KDU, and Senior Lecturer at General Sir John Kothalavala Defence University. I now hand the session over to Dr. Varuna Navaratne and Lieutenant Colonel Dr. Manoj Bandara.
Thank you and welcome all and good afternoon. And we are coming to the fourth and the final session of this IRC 2021 conference. And we have 6 presentations today. And we'll start now. And Dr. Manoj Bandara will present the presentations and the presenters on this conference.
Thank you, Dr. Navaratne. Our first presentation is on in vitro study to identify effects of fetal hemoglobin inducing agents on primary human erythroid cells of beta-thalassaemia major patients. And the authors are N. Yasara, A. Manamperi, and S. Mettananda. The presenting author is N. Yasara.
Greetings everyone, my name is Nirmanya Sara. Today I am here to present the findings of the in vitro study we have conducted to identify effects of fetal hemoglobin inducing agents on primary human erythroid cells of beta-thalassaemia major patients. This is the outline of my presentation. As you are aware, beta-thalassaemia is a monogenic disorder associated with the quantitative reduction of beta-globin synthesis, resulting deposition of excess alpha-globin levels in erythrocytes, which is the main underlying pathophysiology of the disease complications. Unfortunately, the only remaining cure for this condition is hematopoietic stem cells transplantation. However, this is only available for a small subset of patients due to the difficulty of finding a proper donor, high cost, and the risk. However, several studies have concluded high levels of fetal hemoglobin beyond infancy have a protection from disease complications. Therefore, reactivation of gamma globin via potential pharmacological compounds has been a major target in studies focused on discovering new management approach for beta-thalassaemia. Therefore, the objective of this study is to identify effects of selected fetal hemoglobin-inducing agents in vitro in human erythroid cells of beta-thalassaemia major patients. This is a methodology we followed. First, we collected blood samples and isolated mononuclear cells, followed by isolation of hematopoietic stem cells, and we optimized a 19-ray 3-phase liquid culture system to induce erythroid differentiation. After optimizing our protocols, we incubated cells with selected compounds and evaluated their effects favoring globin expression in regard to cell expansion, viability, morphology, erythroid differentiation, and mRNA expression of globin genes. So using a three-phase liquid culture protocol, hematopoietic cells were expanded and differentiated into mature erythroid cells, and cord blood of 3 healthy individual volunteers were used to isolate hematopoietic stem cells for optimization, and cells were grown in IMDM media containing human holotransferrin, recombinant human insulin, heparin, inactivated AB plasma, fetal bovine serum, and penicillin and streptomycin. Depending on the phase of the cell culture, the base media was supplemented with different concentration of heparin, erythropoietin, interleukin-3, and stem cell factor. And the fold expansion, cell viability, morphology, erythroid differentiation, and mRNA expression of alpha, beta, and gamma globin genes were evaluated on day 7, 10, 14, 17, and 19. And cells were maintained at 37 Celsius in a humidified CO2 incubator. Fold expansion of cells was assessed by counting cells under light microscope using hemocytometer, and Trifon blue staining was used to assess cell viability, while Leishman staining was used to evaluate the erythroid differentiation. And the cytocentrifuge machine was used to obtain the mononuclear cell layers we required for the staining methods, and the expression of alpha, beta, and gamma globin levels were measured using TACMA real-time PCR assay. The hematopoietic stem cells of 3 beta-thalassaemia major patients were collected from Adult and Adolescent Thalassemia Center of North Colombo Teaching Hospital, Ragama, and as controls, we collected blood from 3 healthy individuals after obtaining the consent, and cells were treated with selected compounds on day 7 of cell culture and incubated for 72 hours in a humidified incubator, and day 7 and day 10 we evaluated cell expansion, viability, morphology, erythroid differentiation, and globin gene expression. And these are the tested compounds: hydroxyurea, busulfan, vorinostat, and valproic acid. And vorinostat and valproic acids are histone deacetylase inhibitors, and we got the ethical approval from Ethics Committee at Raga Medical Faculty for this study, and we analyzed data using SPSS software. Let's move to results. As you can observe, cells treated with busulfan, vorinostat, and valproic acid shows a significant reduction in fold expansion compared to day 7. These are cell growth shown as mean fold expansion relative to the number of cells counted on day 7. The hydroxyurea treated cells showed the reduction of fold expansion, but the reduction was not significant compared to the negative control in both the healthy volunteers and beta thalassemia major patients. So vorinostat treated cell shows the lower cell viability, and hydroxyurea treated cells in beta thalassemia patients shows the highest cell viability, but that difference is not significant compared to the negative control. And this represented the cellular images captured by the inverted fluorescent microscope on day 7 and after incubating with selected compounds for 72 hours. Similar to the findings of the fold expansion and cell viability assays, you can see there's a high dead cell count in vorinostat treated cells, and there's a high cell count in hydroxyurea treated group and in negative controls. Cells treated with hydroxyurea, busulfan, valproic acid were in at a basophilic erythroblastic stage compared to the controls, indicating these compounds didn't alter cellular differentiation. Let's move into the mRNA expression analysis, and this mRNA expression was calculated in relative to the housekeeping gene, which is RPL13A in this study, and you can see the lowest mRNA expression was observed in vorinostat treated cells, partly due to the low cell viability. And here there was a significant increment in hydroxyurea treated cells in gamma globin expression, and these are the expression values for healthy volunteers. And figure B shows alpha, beta, and gamma globin fold expression related to the negative controls, and hydroxyurea shows the highest expression levels in all three genes. These are the alpha, beta, and gamma globin mRNA expression values in transfusion dependent beta thalassemia patients. You can see compared to the healthy volunteers, healthy controls, these patients are significantly higher alpha globin mRNA expression, and therefore the figure B shows the beta and gamma globin expression related to the RPL13A gene, the housekeeping gene. You can see here valproic acid treated cell shows the very significantly higher gamma globin expression compared to the negative controls. So in conclusion, hydroxyurea did not alter the cell expansion, viability, or differentiation of erythroid cells of beta thalassemia major patients, and in healthy volunteers as well in vitro, favoring its role as a pharmacological agent to induce fetal hemoglobins. Valproic acid also significantly induced gamma globin expression in erythroid cells of beta thalassemia major patients, but it was associated with a significantly lower fold expansion. Actually, this study is an interim analysis of ongoing study. So in the future, we are planning to analyze gamma inducing effect of several other compounds as well. So these are the references I referred for my presentation, and thank you very much.
Thank you very much, Ms. Yansara, for your presentation. Now the presentation is open for the discussion.
That was a really lucid explanation of an elegant laboratory experiment.
Thank you,
but
sir.
I didn't quite catch what the clinical implication of this finding was. Can you elaborate that a little bit?
Yes, sir. So hydroxyurea is actually originally used for as an anti-cancer drug, but after finding its ability to induce fetal hemoglobin levels, it was introduced to the management of sickle cell anemia. Now it's FDA-approved drug in sickle cell anemia management. Then eventually, several studies concluded that hydroxyurea has a very eminent role in, beneficial role in beta thalassemia as well. So with this finding, we can prove that hydroxyurea also has a beneficial role in beta thalassemia, because we did another clinical trial on beta thalassemia patients parallel to this in vitro study, in which we gave hydroxyurea for a 1 year period. Then we found out that compared to the placebo group, hydroxyurea showed a increment in fetal hemoglobin levels as well as hemoglobin levels in beta thalassemia patients.
So they needed less transfusion, etc.
Yes, sir. Yeah, responders needed less transfusion compared to the non-responders and the placebo group.
Right. Thank you. Thank you. That's very clear.
Thank you, sir.
Now, Yasara, when you isolated the hematopoietic stem cells,
yes,
for positive selection, did you check the purity?
No, so unfortunately, at the moment, we got antigens for flow cytometer assays. Due to the COVID situation, we were planning to do it at MRI. So due to that situation, we couldn't get an opportunity to do our study. But after the lockdown ends, we have coordinated with Colombo Medical Faculties. We are planning to do that in future samples.
Because you need to prove that at least it's the purity is at least more than 98% or 99%, and you have to establish the method.
Yes, sir. We have actually used optimizer. We optimized the protocol like from a previous study, using a previous study, and here for cell isolation, we used a magnetic bead cell separation method. So we tagged cells with a magnetic bead like antigen with specific for CD34 cells. Then we pass the cell suspension through a magnetic column, and then we isolated the cells. So in that following that method, we assume that we have a high purity of CD34 positive cells in relative with the evidence guided by previous studies.
What other markers are going to use to check the purity apart from CD34?
28, sir.
What about CD45?
Yes, sir. Yeah, we are doing CD34, 28, and 35. Yeah, 3 antigens.
Any other questions or suggestions?
I'm Professor Navaratne. I am not very clear. Did you do experimental or an intervention on volunteers in this research? No, I know it's a lab study, but at one point in time, I think there was some reference to giving this to volunteers.
No, madam. We did it to actually, this is a part of my PhD study. So I have done 2 studies, one a clinical trial, placebo-controlled double-blind, in which I gave hydroxyurea capsule to patients. And then this is the other part, in vitro study. So in this study also, we tested hydroxyurea. So likewise, the other drugs also currently giving to different indications like leukemia or seizures. So with this help, we can provide some guidance for their use indication to beta-thalassaemia major patients.
So this presentation basically is an in vitro study.
Yes, yeah, madam, which has a beneficial role in management of beta-thalassaemia patients.
Okay, thank you.
Any other? Dr. Balavardhana, can you unmute?
Sorry about that. Yeah. Now, what is the rationale of selecting these 4 drugs? Hydroxyurea, I can imagine that has been used widely, but what is the rationale of selecting other 3 drugs, for this study? Especially like histone deacetylase inhibitors. What is the reason? Because that is the epigenetic inducer. So that is anyway going to disrupt your cell line.
Yeah, so actually I selected these compounds based on literature. These compounds have tested on several studies on in vitro studies, and sometimes they have given to thalassemia patients to check the induction ability of gamma globin induction, but the exact mechanism of inducing gamma globin is not clear yet in these compounds. And one other point is all these drugs has the FDA approval, so they are already we are using these drugs for other indications.
Right. You didn't want to test something like thalidomide on this study, which is a more
yes, actually in the next part, because we have to hold our study due to corona situation, we couldn't continue our cell work. But in the future, we are planning to check thalidomide, azacitidine, and butyrate, and some other selected compounds.
Yes, I think thalidomide should have been tested in this because that is a better proven drug.
Yes, sir.
Okay, in the absence of further questions and suggestions, I would like to thank Ms. Yasara, and also would like to invite the next presenter. The next presentation is on the detection of variably present genotypes, bimABP, bimABM, FHA, B3, YLF, BTFC and LPSA in Sri Lankan clinical isolates of Burkholderia pseudomallei using real-time PCR-based molecular assay. The authors are K. Jayaratne, H.S. Jayasingarachchi, E. Correa, J. Mascorala and A.D.D. Silva. And the presenting author is K. Chairperson.
Hello, good day to you all. My name is Chairperson. Today I am presenting to you the findings on the study detection of variably present genotypes bimABP, bimABM, FHAB3, Variably, BTFC and LPSA in Sri Lankan clinical isolates of Burkholderia pseudomallei using real-time PCR-based molecular assay. So here's the contents of the presentations that I'll be walking you through today. Giving you a brief introduction about bacteria that is associated with this study. Burkholderia pseudomallei is a gram-negative environmental bacterium which is found mainly in soil and water. As depicted in the pictures, can be in paddy fields, other cultivating areas, or in natural water springs as well. So this bacterium is endemic in South and Southeast Asian regions and also considered a tier 1 biothreat organism by the Centers of Disease Control and Prevention, because this bacteria has the potential ability to cause an infectious disease which can be deadly. So that name of the disease is melioidosis. So melioidosis can be potentially fatal, and it was considered an emerging infection in Sri Lanka earlier, but now considered an endemic infection because of the increased number of cases reported every year. So the mortality rates of melioidosis is around 40%, and there's a vast number of clinical presentations in melioidosis, which also makes it hard to diagnose. So the clinical presentations may include pneumonia, skin complications, neurological complications, and sepsis. So the main mode of transmission of this disease is through exposure to contaminated soil or water, or else inhalation of dust particles contaminated with the bacterium, or ingestion of water contaminated with the bacterium are also methods of transmitting the disease. So epidemiology reveals that primarily this bacterium or the disease is reported in Southeast Asia and Northern Australian regions. So talking about the variably present genes of this bacterium possesses several genes which are important to analyze the geographical distribution of the bacterium. They are variably present in these isolates. So examples are Burkholderia intercellular motility factor A, bimA, which has two allele variants, bimABP and bimABM, filamentous hemagglutinin 3, FHAB3 gene, and the two gene clusters here, Yersinia real, YLF, and Burkholderia thailandensis flagellum and chemotaxis gene cluster, BTFC, and the lipopolysaccharide O antigen type A, LPSA gene cluster, which is focused in this study, which has the ability to be used as genetic markers to find the geographical distribution of this bacteria. So the current knowledge on this variably present genes and the ability to being used as genetic markers is not much available in Sri Lanka. So this study is important to determine the prevalence of these genotypes and also to use them as genetic markers. So the main aim of the study was to determine the prevalence of variably present genotypes, as mentioned earlier, in 51 clinical isolates of Burkholderia pseudomallei from Sri Lanka. The main objectives were to perform a dye-based real-time PCR and bell curve analysis to detect the respective genotypes in these 51 clinical isolates. Further analysis was done to the clinical data associated with the patients from the samples that have been obtained from in this study. So this slide represents the methodology that I followed using in this study. So first and foremost, ethics approval has been obtained from the Ethics Review Committee, Faculty of Medicine, University of Colombo, and 51 clinical isolates were used in this study of all from across the country, isolated between the years 2015 to 2018. So the total genomic DNA were extracted from heat-killed cultural isolates, Burkholderia pseudomallei clinical isolates. These 51 clinical isolates were previously confirmed to be culture positive Burkholderia pseudomallei and also confirmed by LPXO, real-time PCR assay to be Burkholderia pseudomallei. So this extracted the total genomic DNA concentration was determined by performing 1% aggregate gel electrophoresis before to the real-time PCR. Then real-time PCR was performed to determine the genotypes for each DNA sample. And melt curve analysis was performed to determine the presence or absence of these genes. As mentioned in the diagrams, these are few melt curve images obtained for each genotype. And the temperatures are the optimized melting temperatures that we use to detect the genotypes. So this table contains the names of the primers, the optimized annealing temperatures, and the optimized melting temperature that we use to detect these genotypes qualitatively, and the sequence of the primer that we used for the real-time PCR assay. So moving on to the results and discussions of the study. First, there's the genetic variation of the bimA gene. There are two added variants as bimABP and bimABM. bimABP is more prevalent in this study population, which is around 76.47%. And also bimABP have been found to be more prevalent in Thailand, which is around 100%, and Australia around 88%. So there's an interesting factor about the bimABM genotype because it has been found to be associated with neurological melodiosis cases in Australia. But in this study population, the clinical data did not reveal any information on presenting neurological symptoms or neurological melodiosis in patients, which has bimABM or in this study population. So this study population is a small study population, and when the sample size increases, the variation of the clinical presentations and the genotypes might change. That is a limitation of this study. So talking about the genetic variation of the YLF and BTFC gene clusters, it is obvious that YLF gene cluster is more prevalent, and BTFC is not much prevalent in the study population. So BTFC is also rarely found in Southeast Asian countries and is considered uncommon in Australian population as well. Talking about the genetic variation of FHAB3 gene and the LPSA gene cluster, FHAB3 gene has showed positivity for around 50.98% of the study population, and LPSA gene type was showing positivity for around 68.63% of the study population. So there's another study that has been conducted, which is under the publication process, which I was also a part of, which had used 310 samples and showed 70% of the population for positivity for the FHAB3 genotype. Another limitation of this study, since the sample size is higher, when the sample size increases, the numbers might change. So FHAB3 prevalence in Thailand and Australia have also been found as 100% and 83% respectively. And LPSA gene cluster is considered to be highly prevalent in Southeast Asian isolates as well. Talking about the clinical data associated with the patients from which these samples have been identified from, the graph here, the histogram, represents the age distribution of the patients from which these samples have been identified from. So melioidosis patients have been identified from less than 10 years old to less than 90 years old, which is a vast age distribution for melioidosis patients infected in this study population of 51 cases. So this pie chart represents the outcome of the patients which the clinical sample have been obtained from, whether they were recovered, relapsed, or deceased. So an equal percentage have been deceased and recovered, which is around 47.06%, and 5.8% of the population had reported a relapse. This graph here represents the risk factors associated with the deceased and recovered patients, as to which what are the risk factors associated with the deceased patients and what are the risk factors that are associated with recovered patients and in what percentages they are in. So it is clear that both the deceased and recovered patients had demonstrated risk factors, but diabetes is the highest percentage that is observed here. And Cushing syndrome is the risk factor that has been observed in the lowest percentage. Concluding the presentation, I would like to state that majority of the clinical isolates in this study population demonstrated YLF gene cluster, and bimABP genotype. But when sample size increases, this may change. So the Sri Lankan population type is a mixed type population. So majority of the population did show bimABP and YLF genotypes. And the mortality rate of the patients in this study was around 47.06%. And diabetes was found to be the risk factor contributing towards the mortality in the highest percentage. And being diagnosed with Cushing syndrome was the lowest contributing factor towards mortality. Both deceased and recovered patients demonstrated risk factors in this study population. So these are the references that were used. Thank you all for listening.
The presentation is now open for discussion.
Again, I want to ask you the same question I asked the previous speaker. Now, yours was essentially a laboratory study. How does this laboratory study actually influence either diagnosis or management of the patients? You gave some clinical correlates, but that was not related to the laboratory findings. How does your identifying these the laboratory findings that you, how will it help either in diagnosis or in management? Did she hear me or not?
Ms. Jayaratne,
Yes.
can you please unmute?
Yes. Yes. Sir. So the diagnosis of this disease is done at the University of Colombo Medical Faculty for melioidosis. So this study was mainly to identify the genetic markers that could be used as to check the geographical distribution of the disease. This could be further analyzed with the associations of the clinical presentations of the disease of the patients and the presentations of the genetic factors, and further analyze the associations to find out whether there are any associations with the presentations of the genetic factors and the disease outcomes as well.
But your sample is too small to come to any such conclusion.
Yes, sir.
What you're saying is there may be implications about the genetic thing which influence outcome, is it?
Yes. So there's another the bimABM genetic marker that was also used in this study has been found to be associated with presenting neurological melioidosis in other countries. But this study population did not show any clinical data that reveals presentations in these 51 isolates.
Okay, thank you.
This is Darshan. If I can just add to that briefly, with this disease, this has been studied very little in Sri Lanka, and even globally they are having trouble actually, connecting these correlates. So in Sri Lanka, as you see, we are still losing half the people who have been infected. So we have actually, and there is a treatment for it also. You have to give meropenem IV and all that. So we are still at a very early stage of detecting this disease and getting there. So we are trying to basically get the basics together first, and then hopefully we'll be able to, I mean, your question is very relevant and very important. Unfortunately, funding delays and all of that has delayed this. So it's a question we hope to answer, and we are trying to get to that answer what you asked.
Thank you.
Thank you.
Thank you.
Any other questions and suggestions?
Yes, I want to know whether you would call this a newly emerging disease, or the other countries you quoted like Australia, etc. Have they had diagnosed and has got a high prevalence? What is the situation? Is it something that is fairly low prevalence, and I don't know, for whatever reason it has not been diagnosed? Can you tell us a little bit more about actually why this is known so little about, and why there's such a high mortality, right, case fatality also?
Yes, madam. This disease is considered endemic in Sri Lanka right now, and it is considered endemic in other parts of the world like the parts of the world like Southeast Asian and Australian regions as well. Earlier, this disease was considered an emerging infection in Sri Lanka, but with the increased number of cases being reported, now it is considered an endemic disease. Since the clinical presentations is so vast and the variety of clinical presentations of this disease is very high, it is being hard to diagnose this disease at once. But now with the National Surveillance Program in Sri Lanka, all hospitals are, I think, made aware about the clinical presentations and this disease. So the high number of cases are being reported in Sri Lanka right now.
Dr. Himali, do you want to add to that, or shall I say something? Madam, to just answer, you can read Darshan again. Basically, until something like 2012, this disease has been around for a long time. This was first discovered 1911 or so, and then like the third case was reported in Sri Lanka. After that, there's a huge gap where people just lost interest over it. We just didn't know about it. And then again around 2000 or late 90s, then Professor Vasanthi Tevanesam did some studies and found. Then Himali, did you want to add to this?
Yeah, actually, in coming presentation is there. I think there will be another presentation about the melodiosis and the Burkholderia pseudomallei. So we can explain that actually right now cases are reporting around the country and clinicians are more aware of that after surveillance program also. And now I think cases are increasing. More cases are reporting from the country by now.
Okay, thank you. We'll listen to the other one as well.
Okay, while thanking Ms. Jayarathne and the team members, let us move to the third presentation. That is the conservation of in-silico predicted epitopes of SARS-CoV-2 and other native coronavirus from different geographic regions. The authors are ATAD Karunatilaka, NPDS Lasanthi, CL Gunasekara, and SS Seniviratne. The presenting author is ATAD Karunatilaka. It's over to you.
Good afternoon, everyone. I am here to present about my research findings on conservation of in-silico predicted epitopes of SARS-CoV-2 and other native coronaviruses from different geographic regions. To give a brief introduction to this topic, COVID-19, which was probably originated in Wuhan, China, now has infected over 290 people, killing nearly 4.55 million as of September 2nd. The coronaviruses are known to be the largest RNA viruses, and they can be categorized into 4 different groups, namely alpha, beta, gamma, and delta. There are 7 known human coronaviruses, including SARS-CoV-2, and all of them are of zoonotic origin. When we look at the genome structure of SARS-CoV-2, it has a 5' cap structure and a 48 tail at the 3' end. The gene order is as follows, as indicated in the part B of figure 2. There are 3 conditions that should be fulfilled in order for a zoonotic spillover event to happen. The first one is the establishment of a host reservoir of coronaviruses, and second is the establishment of direct or indirect route of transmission between the host reservoir and the recipient. This may happen via 3 ways, namely the consumption of viral hosts by the humans like bushmeat, host viral shedding, as well as the tissue tropism coronaviruses through the close interaction between the target tissues of the recipient and the host guano. The last factor that should be fulfilled is the presence of suitable recipient hosts in the areas where this viral shedding is taking place. The shedded viruses can bind efficiently if there are highly conserved host receptors. The immune-informatic epitope prediction of SARS-CoV-2 proteins helps us to identify the antigenic epitopes. This mainly helps us to understand the different reasons behind the varying mortality rates in different countries, as indicated in this figure 4. It also helps us to give an understanding on the variable nature of symptoms in different individuals, which is varying from asymptomatic, mild to severe symptoms. When we pose the question, why is it important to identify the cross-reactive homologous epitopes of coronaviruses of different geographical origin? As we all know, the SARS-CoV-2 proteins can be divergent from the endemic H cores as well as from the zoonotic cores. However, there are highly conserved regions or homologous regions between these coronaviruses, and this may open up the potential or chance for an immune cross-reactivity, and it can be solved only at the epitope level. If an individual is exposed to a previously closely related coronavirus infection, it can activate the memory T cells in that individual, even though these epitopes may have minor genetic differences. So this would create potentially different degrees of immune responses in different individuals. It also helps us to understand and properly interpret the results of different serological studies, such as serosurveys or clinical antibody tests. The hypothesis drawn for this study was the epitope conservation between SARS-CoV-2 and other native coronaviruses, including SARS, MERS, endemic H cores, and zoonotic cores would create a cross-immunity in the endemic regions. To brief about the method section, the first step is the acquisition and the processing of sequence data, under which the human co-isolates as well as the zoonotic cores are downloaded from the NCBI GenBank. Under the zoonotic cores, paleo and neobat cores, pangolin, palmsnet, and mink cores are downloaded, and geographical regions of these zoonotic cores are indicated in this map. The second step is the epitope prediction, under which the B and T cell epitopes are predicted, and under the B cell epitope prediction, linear and discontinuous epitopes are predicted for the four structural proteins of the genome using different softwares. Under the T cell epitope prediction, the MSc class 1 prediction was done for the 8 most frequent HLA class alleles with phenotypic frequency greater than 10% in the worldwide population. The predicted epitopes were subjected to categorization, and the top 1 epitopes was selected. Under the MHC class 2 epitope prediction, the IEDV median consensus percentile of 20% threshold was used, and the predicted epitopes were subjected to antigenicity evaluation using Blackgen 2.0. The third step is the epitope conservation analysis, under which the H-code isolates as well as the zoonotic cores are compared separately with the predicted reference SARS-CoV-2 isolates, namely linear and discontinuous B cell epitopes and MHC1 and MHC2 T cell epitopes. These predicted epitopes were used to generate graphs using the ggplot software, and the tables were separately generated with a conservation rate for each individual epitope. To summarize the results, the linear B cell epitopes, which are predicted for the structural proteins, as they are the accessible antigens for the B cell receptors, the different types of antigenic epitopes are predicted as indicated in this table. Under the discontinuous B cell epitope prediction, as indicated in figure 4, the spike region has generated 25 potential epitopes, whereas the nucleic acid region has generated 51 potential epitopes. Under the MHC1 T cell epitope prediction, a total of 330 antigenic epitopes were predicted belonging to 9 different protein regions, as indicated in this table. The figure 5 gives one such example, which is the MHC1 antigenic epitopes of the spike region. When we look at the MHC2 T cell epitope prediction, similarly to MHC1, a total of 201 antigenic epitopes were predicted for the 9 different protein regions, as indicated here, and the figure 6 gives the antigenic epitopes of the spike region. The phylogenetic tree, which is a maximum likelihood tree developed using the coronavirus sequences, which were compared to the SARS-CoV-2, as you all can see here, the closest relative of SARS-CoV-2, which is the RATG13 CoV, which is a bat CoV from Rhinolophus actinis bat, forms a cluster with the SARS-CoV-2. This cluster forms a separate clade with the predicted intermediate host of SARS-CoV-2, which is the pangolin CoV. When we look at the Rhinolophus bat CoVs of China, they form a separate clade, and they are also closely related with the SARS-CoV-2. When we focus on the SARS and its intermediate host, which is the palmsnet CoV, they are forming sister branches of the same clade, and they are also closely related with the SARS-CoV-2. However, when you look at the alphabet codes of the New World, which means the North and South America, they are forming a separate clade, and they are very distantly related with SARS-CoV-2. When we focus on the human coronavirus, both alpha and beta codes form separate clades, and they are distantly related with SARS-CoV-2. However, we should pay more attention to these Rhinolophus bat codes in China, because they are established reservoirs of SARS-related codes. Further, they have a vast geographical distribution, which is varying from Europe, Africa, Asia, and Australia. This table gives the number of epitopes which are showing 100% conservation under different proteins of the different cores. As you all can see here, A and L, which gives the Rhinolophus aphanis bat cores and pangolin cores, they show the highest number of 100% conserved homologous epitopes across different proteins, whereas the Rhinolophus bat cores as well as the pangolin core and the SARS-CoV also show a moderate number of 100% conserved epitopes under different genomic regions as indicated in this table. The figure 9 gives a schematic representation for this conservation of MSC1 epitopes, and this is one such example where the y-axis gives the conservation percentage, whereas the epitope number is given on the x-axis. The data point color intensity increases when the number of overlapping sequences increase for that particular conservation level. As you all can see here, under the epitope number 1, it is 100% conserved across the Rhinolophus bat cores, the pangolin cores, palm civet cores, as well as from the SARS-CoV as well. This gives the conservation of MSC1 epitopes across different proteins, and it also follows the same pattern as I indicated before, which is the Rhinolophus aphanis bat core or the RA313 showing the highest number of 100% conserved epitopes, followed up by the pangolin core and the Rhinolophus species bat cores. When you look at the SARS isolates also, they also show a considerable number of 100% conserved epitopes across different genomic regions as indicated here. And the human coronaviruses as well as MERS coronaviruses show low to moderate conservation. However, the alphabet cores and the mean cores show very low conservation. The figure 11 gives the conservation of MSC2 epitope conservation across different proteins, which is similar to MSC1 conservation as I indicated before. The figure 12 gives the schematic representation of peaceful epitope conservation across the structural proteins, which are 4 structural proteins, and these are predicted for the 2 different types of diseases, which are linear and discontinuous. The conclusions drawn for the studies are the SARS-CoV virus shows a higher conservation level across a number of epitopes, while the endometric cores show moderate to low conservation across different epitopes. The average conservation may range between 30% to 50% in both B and T cell epitopes. When we look at the percentage epitope conservation of SARS-CoV-2, it is significantly higher in zoonotic cores such as the Rhinolophus bat cores and the Malay and pangolin cores. So, these results suggest the possibility of an existing remnant immunity in individuals who are living in the areas where these Rhinolophus bats and the Malay and pangolins live, such as China being the hotspot, followed up by the South and Southeast Asian countries due to the exposure to the zoonotic cores over a long period of time. This conclusion is postulated only based on the very high level of epitope conservation between SARS-CoV-2 and these zoonotic cores. These give the 3 references of my study, and these are the people I should acknowledge for the success of my study. Thank you.
Now it's a time for questions and suggestions.
As no one seems to be asking a question, I will again ask now here. First of all, you are not talking about COVID-19, but other pathogenic COVID viruses, from I understood, and you are implying that these epitopes would determine a relationship to an animal host and the immunity. So can I ask you in practical terms in Sri Lanka, what are the potential animal hosts for seriously pathogenic COVID viruses to be hosted?
So there's a current study carried out by the University of Colombo regarding the bat coronaviruses available in Sri Lanka, and there's also a published study which was done for the Pteropus medius bats, and these bats are showing to host beta coronaviruses, which is the same type of viruses which are similar to SARS-CoV-2, and they are just 5.8 to 6.7% different from the SARS-CoV-2. So which means that they are very close together. However, there's no evidence yet whether they are infecting the humans directly. For that to happen, the host receptors and the bat receptors should be similar. So there's no such evidence yet. but the current studies are going on, and people should see whether if we check the individuals, if we check the serological studies, if we carry out a serological study to such as to look for cross-reactive epitopes from the serum samples from individuals who are living in these areas where these bats live, we could get an idea whether these bat cores provide a cross immunity to the SARS-CoV-2 infection, which is COVID-19. So that's my idea.
Thank you. You know, are a wide species, you know, variety of species like say polecats and mongooses and all, you know, there are some which inhabit houses. There are potential hosting ability. Is it being looked at by some groups in Sri Lanka?
Not yet, sir. Because for SARS virus, the palm civet was the intermediate host, and in Sri Lanka also civets are available in house gardens as well. So far, there's no study carried out for these animals, but there is a study currently carried out for the different bat species living in Sri Lanka, especially the Chairperson medius bat, as well as there are certain bat species called Chaperon plicatus, and it shows a higher conservation level, or as 100% high number of 100% conserved epitopes in my study as well. And these bats are found in Sri Lanka as well. So if we study those bats and check whether there is a cross-reactivity in the antibodies, we could come to a conclusion whether there is a possibility of these bats infecting humans as well. For that to happen, several conditions should be maintained. As I mentioned earlier, these host receptors should be similar to the bat receptors.
Thank you. Thank you.
Can I ask a question now from the presenter? Is there any evidence to say in endemic regions of rhinolopus bats and the pangolins, any cross-reactivity with SARS-CoV-2 and these zoonotic viruses?
Yes, sir. There was a study carried out in Thailand. They have checked in 2003. They have checked the pangolins. Pangolin is the predicted intermediate host for SARS-CoV-2, and they have found serum samples from this Manis javanica or this pangolin, as well as samples from 2020 in Thailand. They also have neutralizing antibodies against the SARS-related cause. So it means that these viruses are hosted in those species over a long period of time. This gives a potential time frame for these coronaviruses to be infected to humans. And in bat species like Rhinolopus affinis and Rhinolopus siniscus, they also use the same receptor like humans for the bat cause. They use the S2 receptor for the binding of coronaviruses, and so these are the available literature for the possibility of coronaviruses circulating in different species.
Yeah, and is there any evidence say that there's some kind of immunity from these viruses for SARS-CoV-2?
Yes, sir. A study was done in China. They have found they are not actually infected at that stage. Now, when they check the serum samples, they have found that from like 0.6% of that population cohort, they contained neutralizing antibodies for SARS-CoV-2 related coronaviruses. So, which means that these bat coronaviruses have already infected those individuals in the areas where the zoonotic coronaviruses reside in bat populations, and there's a higher chance if we check those human populations, especially in countries like China or Southeast Asian countries, as well as we can do it in Sri Lanka as well, because there's a huge population of bats in Sri Lanka, also to confirm this prediction. Here I have predicted this only using the epitope conservation levels.
Right. Thank you. Any other question, please?
Okay. Thank you very much, Ms. Karunatilaka, for your presentation and also for clarifying the doubts of the audience. With that, let us move to our fourth presentation, that is molecular characterization and phylogenetic relationship of SARS-CoV-2 strains infecting a Navy cohort during the first wave of the COVID-19 in Sri Lanka. The authors are H.S. Jayasinghe Arachchi, D.S. Govindapala, WMID Nakavita, PH Premaratna, CL Gunasekara, DGP Kavyangana, KWT Chaturanga, US Kulasekara, and ADD Silva. The presenting author is HS Jayasinghe Arachi.
Good evening, everybody. I'm going to present our molecular epidemiology study of severe respiratory syndrome coronavirus 2 infected in asymptomatic, minimally symptomatic COVID-19 patients in Navy cohort in Sri Lanka. My presentation contains introduction, materials and methods, research and discussion, conclusion, references, and acknowledgement. Severe acute respiratory syndrome virus SARS-CoV-2 is a causative agent of COVID-19 pandemic. It was emerged in December 2019 in Wuhan, and it's a single-stranded positive RNA virus having genome size 26 to 32 kb in length. It belongs to beta coronavirus. And 96.2 genome similarity was found with bat coronavirus obtained from Yunnan province of southern China. So SARS-CoV-2 positive RNA extract have been used for the molecular epidemiology studies to reveal very important information on mutations and fast evolving strains and transmission routes. So this study is based on the first 26 asymptomatic, minimally symptomatic males from Navy COVID-19 patients diagnosed with COVID-19 by real-time PCR. Our aim of this study was to investigate molecular epidemiology and phylogenetic relationship of SARS-CoV-2 strains infected in Navy cohort. And we want to compare with the other globally circulated strains during that time, during the first wave. So objective of study was to find the distribution of nucleic acid substitutions in 10 genomes, and also to perform real-time mutation analysis, and also to find phylogenetic relationship of these Navy strains to those that reported from locally and globally during first wave. So we have RNA extracts, 10 RNA extracts based on the CT values, high CT values. We subjected to whole genome sequencing. Sequencing was done outsourced. We had to limit for 10 sequences in this study because financial restrictions also there. Ethical approval we obtained from KDU Faculty of Medicine. So we have large number of whole genome raw sequence data, and also we have whole genome data from genome GZ database. So analysis of whole genome sequences we have conducted here. A real-time mutation tracking, we want to do a variant analysis at RNA level, and also we want to perform lineage analysis. Finally, we performed phylogenetic analysis using some genomic sequences. Example, 62 genomic sequences we use for our phylogenetic analysis based on the mutation profiles. We downloaded all data from GZ website. It has all data, and even NextGen is one of the interesting database. That large number of whole genome data were present in this. So variant analysis at the genome level, we used a detective coronavirus typing tool and core server hosted by the GZ server. Phylogenetic and lineage analysis was conducted in phylogenetic assessment of named global outbreak lineage pangolin website. So results of this study, we detected two lineages, B1 and B1.3. So B1 contain only one strain, B1.3 have 9 strains. So we gave the names, strain names we have given according to our lab identification number. These are the accession numbers we have used, given. Actually, the common known mutations, that is amino acid changes we found in our all 10 strains. Those mutations were also found in globally circulated SARS-CoV-2 strain during first wave. These are the list of those common amino acid changes. So our strains are genetically diverse because some of the mutations we couldn't see in all strains. For example, P275 is a mutation in non-structural protein 14 was not found in all the strains. Only three strains carry this mutation. Likewise, you can see here all other mutations. So this slide shows you the distribution of D614G mutation. This is a spike protein mutation. And along with this mutation, this non-structural protein tree Q57H, T851, and N2, P23L concurrently occurred in globally circulated strains. This D614G mutation was emerged in Europe during February 2020, but within a month of time, it spread all over the world, having 70% of the globally circulated strain. Especially in USA strains, most of the strains are carrying this DG614. We analyze phylogenetic relationship. We want to see the phylogenetic relationship of our Sri Lanka existing strains. Few strains have been reported. So we found there are 3 strains reported Sri Lanka during March, phylogenetically close to our strain. Our strain form a different cluster, but we wanted to know whether these are the strains that cause Navy infection. But how we can see, we analyze the mutation profile of those 3 strains. So we found all strains carrying D614G, but not other linkage mutations. Therefore, this strongly suggested that transmission route of SARS-CoV-2 strains in Navy-acquired COVID-19 is not this strains. This may be a different source. And further, we wanted to see, because there are a few others lineages are also circulated during that period, especially lineage B11 and lineage B47, and also B.4. So we have done mutation analysis of those strains also. Those strains were reported before the Navy case cluster emerged. So we found that shared mutations are only found in B11 lineage, but only 2 mutations, not all other linkage mutations. Even B.4.7, we have no found any shared mutations. Therefore, SARS-CoV-2 reported from this study is from different source. B.4 lineage also reported in Sri Lanka during March, but there's no common mutations with our strains, except one mutation. So in phylogenetic relationship also, we found our strains clustered in one cluster, along with the Sri Lankan 3 previously reported SARS-CoV-2 strains and also USA strains. So we first reported B13 lineage from this study, and parent lineage B1 is available. Those strains lineages are very common, predominantly prevalent in USA during that period. So in conclusion, in this study first reports B13 SARS-CoV-2 strain circulating in Sri Lanka during first wave. We found linkage mutation along with the D641G was prominent, which was the prominent lineage clad in USA during the first wave. And also we found B1 lineage having those mutations. Same strains have been found in China during April. So in addition, several other amino acid changes have been observed in our strains, but not in previously reported strains in Sri Lanka. So therefore it can be suggested that the SARS-CoV-2 strains found in this Navy class cluster is closely may be related to SARS-CoV-2 isolate from USA, lineage B13 and lineage B1. So it can be assumed that route of transmission may be linked to some other source, probably because of associated with the travelers from USA. During that time, there were no that time means March, first 2 weeks of March, there were no quarantine regulations imposed our country, but many travelers have come from foreign countries. Thank you.
The presentation is open for discussion now.
I'll start off again. I just want to ask one question. Are your findings consistent with what Dr. Chandima Jivandara told us yesterday about sequencing, or is there a difference in your findings?
So actually, I didn't hear the Dr. Chandima Jivandara's discussion anyway yesterday, but this is actually the strains from first wave. There were few strains reported at that time. So we wanted to know how
no, this is the first wave. I mean, this is from the Navy cluster, isn't it?
Navy cluster during April. Actually, we detected 23rd. April 23rd. So at that time, there were few strains only Sri Lanka. That also hardly 9 strains reported from Sri Lanka at that time. So those strains are not related to this, our Navy strains actually. And mutations actually early evolving period of SARS-CoV-2. By now, there are many evolutions now. SARS-CoV-2. Some of the mutations still existing, like G614G is still existing, and P32L and capsid protein 12 that is related to the high transmissibility is still there in variant of concern nowadays finding. Actually, it's a fast evolving strain. So some of the mutations have gone actually. So.
But I mean, I think you should know whether your findings are consistent with what was presented from Jambiga. You know, those are influencing policy making in Sri Lanka. So it's your duty to see whether it is compatible or you are telling something different.
No, I actually say how can we say a different thing? Because what's the data available in the GSET? Actually, they also have uploaded the data available in the available GSET. Based on those data only, we are making conclusions. Our data also very accurate, and those data their data also we are using at that time. During that time, very few sequences. This is actually a story of the first wave, not related to now.
So this is Darshan. If I can just add, Nilika does not have strains from the samples from this time. I actually talked to her. So this is actually the first strains because they only started sequencing after this. They did not have the samples from this wave. So this is the first set actually that is available in Sri Lanka.
You mean USJ has not done any samples from this particular cluster?
No, sir. She was not able to. I talked with Nilika about this because they didn't have any samples available from the Navy cluster because they only got organized and they started after this cluster because you had to preserve it properly. Now this first cluster came to our lab and we immediately stored it at -80 and we had that stored. Unfortunately, because of funding issues, it took us a long time to sequence it. We could only do it about a year later because we didn't have the funding. But Nilika sequenced only after the Navy one. So our ones are actually, the first ones to get sequenced here. We need to publish this soon. Yeah.
Actually, based on the detection isolation date only, we are reporting. If Professor Neelika has a strain, she could have reported by now, but there were no data in the GZ or NCBI side. So based on the available data only, we are making conclusions. So by now there were no data. Some data available from their laboratory actually. Anybody can go and see GZ site. Most of the regions are in. This is no identification of any bases. Some issue in the sequencing actually. So how we can see the mutations present or not, we can't say. Anyway, based on the available data only, we are making some conclusions here. So I don't think any inconsistencies will happen actually. Thank you, sir.
Okay.
Yeah, can I ask a question now?
Yes, sir.
Dr. Jaisingh Arachi, now what you have presented is in Sri Lanka is the first time that the sequencing have occurred in this COVID outbreak. This is the first time it has been done. First isolated in Sri Lanka. Is that correct?
This is the first. No. Before that, Jawadharpur University has started the sequencing work before us.
No, before April, have they done any sequencing?
Yes. 9 sequences are available.
Okay.
Varuna, just to clarify now, what I said was from this cluster they didn't have samples. That is what. Yeah. So from this cluster, basically, this is the first one. Yeah.
Any other comments?
I think.
So I would like to thank Dr. Jaisingh Arachi and the rest of the members of the team. And let's move to the 5th presentation. That is the identification of new variant of SARS-CoV-2 virus of C36 lineage with L452R mutation at the University Hospital KDU, Sri Lanka. The authors are H. Abhegunawardana, UTN Seneviratne, MMPT Jayasekara, WMID Nakavita, FN Mubarak, US Kulasekara, DGP Avyangana, DS Govindapala, and ADD Silva. The presenter is H. Abhegunawardana. So over to you.
Good morning to you all. I'm Harshi Abhegunawardana, a technical officer at the Faculty of Medicine, KDU. Today, I would like to present a case study on identification of new variant of SARS-CoV-2 virus of C36 lineage with the L452R mutation at University Hospital, KDU, Sri Lanka, which is very relevant in the present context. The COVID-19 pandemic has affected the lives of all of us, making the year 2020 and even this year nothing like we have ever experienced before. The freedom of movement has been greatly limited, while the public is closely following the news to find out the latest developments on the COVID-19 situation. But it is hard to understand the variants, mutations, and so on. There are many variants of SARS-CoV-2 circulating around the world with new mutations identified regularly. Therefore, it is important to closely monitor the emergence of new variants of SARS-CoV-2, which can further spread the virus. Here, I will describe a new variant we discovered from a family admitted to UHKDU. This is the structure of the virus, and the green globes shown here are the spike protein, which binds to the ACE2 receptor. The enlarged figure shows the S protein and the receptor binding domain. As you all know, the mutation can occur in these spike proteins. The next image also the various locations where mutation have occurred over time. As a result, you have the various mutants. Laboratory studies suggested that the cases of COVID-19 caused by variants with certain substitution or combination of substitutions in the spike protein may be more resistant to the vaccines. The US government SARS-CoV-2 interagency group developed a variant classification scheme that defines 3 classes of SARS-CoV-2 variants: variant of interest, variant of concern, variant of high consequence. The alpha, beta, delta, and gamma variants circulating in the United States are classified as variants of concern. No variants of high consequence have been identified to date. As the pandemic continues, variants have become the latest concern, with notable examples of variant of concerns are emerging. These variants are complicated. Each one is made up of a collection of mutations as shown in the previous slide, all of which have the potential to change the SARS-CoV-2 virus in unexpected ways. Viruses multiply by copying their genomes over and over. But like an old photocopier, these copies are not always perfect. Each of these imperfect copies is a variant. These are the variants which have been identified previously in Sri Lanka. According to our case study, a total of 6 members of one extended family were infected with SARS-CoV-2 over the 5 days. Short time frame of infection with high number of cases, suspicion of the same strain of virus was considered. Index patient P1 and his father P2 got symptoms at the same time. His mother-in-law P4 could have been infected by traveling in the same car from Galle to Colombo with the index patient, and P3 could have been transmitted by her husband. P5 may have got the infection from P3 or P4, or could be P6. By the time of P3 got positive, P5 was negative. Therefore, it is impossible to transmit the virus from P1, which is index patient. P6 got from P3. This is the timeline of index patient, and L indicates the living with index patient, and C indicates the contact for other family members with the index patient. S indicates the developing symptoms, and H indicates the time of admission to the hospital. These RNA samples of P3, P5, and P6 were recovered from the Molecular Diagnostic Laboratory UHKDU and sent for SARS-CoV-2 NGS sequencing. Sequences were aligned using Genius software and phylogenetic tree developed using Genius Tree Builder using the neighbor joining method. The SARS-CoV-2 variant found by UHKDU Sri Lanka was found to be in the Pangolian lineage C36, alias of B11136, mainly from Egypt and other countries. While the variant mostly occurred in Egypt, 33%, Germany 12%, United Kingdom 11%, United States of America 8%, and Denmark 7%. The significance of this mutation, L452R, has been studied by several research groups, and our case study variant is compatible with Oman strain. While the D614G present in all of the strains on this tree, L452R, is present in all strains below France strain. Considering this tree, it is most likely the strain came to Sri Lanka from Oman or that surrounding region. We have submitted our sequences to GIS8 database, which is a European database enabling rapid and open access to epidemic and pandemic virus data. And also we have submitted to the gene bank. These sequences belongs to C36 lineage, although it has a L452R mutation, which is not common in the lineage. And also this mutation is common among B1429 California strain and B1617 Indian variant. This mutation alters the dynamic in the receptor binding motif of receptor binding domain with high infectivity. This is a spike protein mutation. And this is the first report identify a new variant of the C36 lineage containing the L452R mutation from Sri Lanka, followed by detection of the same lineage from Varuna in June 2021. This strain shows close resemblance to the strain sequence in Oman, and we speculate it arrives in Sri Lanka from a traveler from that region. But at this time, this is not a variant of concern, but maybe a variant of interest due to the L452R mutation. Many more samples and a higher spread of this variant would need to be discovered before this variant is highlighted as a concern. The emergence of such variants through the accumulation of convergent mutation or from travelers coming into the country needs further evaluation. Constant monitoring of the spread of known variants of concern and the new Sri Lankan variants are required to determine the impact on public health in Sri Lanka. This slide shows the regional distribution of variant of concern and variant of interest in submitted sequences. This is the distribution of variant of concern and variant of interest in Asian region, which illustrates the changing pattern of Delta variant in the region within short period of time. And these are my references. And I would like to thank Molecular Diagnostic Laboratory UHKDU for conducting the PCR and providing RNA extractions, and BML2 KDU grant for funding the NGS sequencing, and Gene Labs Medical for carrying out the sequencing, and Dr. Himalayas Jayasingharachi for her support for the initial analysis. And thank you.
Okay, the presentation is open for discussion now.
And the clinical and the public health implications of finding variants like this and how the variants are disseminated. It's very easy for me to understand it. Thank you.
Can you hear me, Harshi?
So
thank you
some
very much for
echo is
that.
going.
Can hear you, Prasad.
Yeah, thank you very much for the presentation. Just a very basic question.
So some echo is there.
Identified the disease strain first time in Sri Lanka. Did you able to trace back and see how, I mean, the origin of this sample in Sri Lanka? Any idea about how, I mean, the family got this strain? Any idea about that?
Sir, can you repeat it once again, sir? I can't hear you properly. Some echo is there.
This is the first time that this new strain identified from Sri Lanka. So is there any idea how this came to Sri Lanka now? Were there any, I mean, when you trace back how the first person of your cluster got this disease? I mean, from where, is there any idea about that?
Actually, no, sir. We couldn't found how it comes to this family.
You said that it might have come from Oman. You said something.
Yes,
like that?
yes, sir. According to the phylogenetic tree, we are speculate that it comes from Oman, but we couldn't say how this family got this infection, because no one in the family is no one in this family is not got abroad to this country, this region.
So after that, this strain was again identified from Varuna, no?
Yes, sir. Yes.
Okay, thank you.
And throughout your presentation, the highest degree of clarity prevailed. Good performance and congratulations. Thank you.
Thank you, sir.
Arshi, I also want to say the same thing what Navaratne said. It was a very clear presentation, very crystal clear, and we understood every bit of it. So well done.
Thank you, sir.
Can I ask you one final question now? You said this is not a variant of concern, but at the same time you said it has very high infectivity. So why do you say it is not a variant of concern when there is high infectivity? Any reason for that?
So to consider as a variant of concern, we have to sequence more samples from this strain.
Right.
That's why we couldn't say at that time this is the variant. This is goes to variant of concern at this time. Sir.
Right, but it is highly infectious.
But,
right?
Yes.
Right. Okay.
The mutant L452R mutant is also present in this Delta variant as well.
Right.
So it's highly infected.
This is Darshan here again.
Yes, Darshan, explain that. Can't hear you, Darshan. We have lost the sound.
Darshan, can you hear?
No, I think we have lost the connection to Darshan.
Is there any other questions?
I think Darshan was trying to say that there is a specific criterion for calling it a variant of concern, isn't it? The WHO has very specific.
Yes, sir. Yeah, Darshan, I just came back, sir. If I can just get a minute to, that's actually very, thank you. That's a very relevant question what Dr. Balawardhan asked, and we've been very careful about how we define it. Even when we first found this, we had many discussions and we were worried about this, but then the health minister said, no, you can't go public with anything of this, because the thing is actually, even though theoretically now in the lab, yes, this is very infectious, but we don't know enough about the virus to say how it will actually perform in people. Now, if you've been reading the news recently, now the Delta variant has taken over, and they are saying there's a new variant, the Mu variant, which has come up, which supposedly evades vaccines even more. But the thing is that has not been able to break through and actually take over, at least up to date. So we have to be very careful with this. So even though this looks infectious, now you haven't obviously heard of this strain again. So it has been very limited in how it can spread. So even though we say it has this specific mutation, in biologically, it actually not be maybe not that strong enough to go about and spread, and not as strong as the other strain. So as far as we know, only 2, however, 1 within the family, and then some other case has been reported. Of course, we are not doing a lot of sequencing, so we don't know. But for this to really become important, it would have to spread much more than this. So as Harshi said, yeah, that's it. Yeah. Thank you.
I suppose that is because the transmissibility may depend on not only one single mutation, but maybe several factors. And so the dominant strain has now become the Delta variant because of all these factors are there in the Delta variant, and probably in future the Mu and the whatever else comes like Lambda.
That's right. Yeah, what caught our interest was the fact that this spread among this family very quickly. So that is how my interest got piqued, and then we somehow managed to sequence it. But then, of course, it didn't seem to spread much beyond that. So we just, yeah, but other than the Varuna case, we haven't heard about it. That was sequenced by Neelika, and after that, she hasn't reported this again.
Yes.
Yeah, okay, thank you.
Any other questions?
Okay, in the absence of further questions, I would like to thank Ms. Harshi for her excellent presentation, and also for the members of the research team for their work. With that, we have come to the last presentation of this 4th technical session. That is the unrevealing the genetic diversity of Burkholderia pseudomallei in Sri Lanka using multi-locus sequence typing. The authors are K. Jayarathne, H.S. Jayasingarachchi, E. Correa, A. Fonseca, T. Mutugama, J. Mascorala, and A.D.D. Silva. The presenting author is H.S. Jayasingarachchi.
Good evening, everybody. I'm going to present our study on unraveling the genetic diversity of Burkholderia pseudomallei in Sri Lanka using multilocus sequence typing. Content of my presentation: introduction, materials and methods, results, discussion, conclusion, references, and acknowledgement. Burkholderia pseudomallei is the causative agent of melodiosis. Melodiosis is a potentially fatal infectious disease, which is also known as white moth disease. It was first isolated in humans from a patient with the glanders-like disease by pathologist Alfred Whitemore in 1927. The disease is endemic in Southeast Asia and Northern Australia. The case fatality rate varies from 20 to 40 in other endemic regions. In Sri Lanka, the case fatality rate is around 24%. This map gives you an idea about global epidemiology of melodiosis. You can see highly endemic regions, endemic areas, sporadic cases were reported from the world. So this is in Sri Lanka. Cases are increasingly reported from the country. Most cases are sporadic. Case clusters also occurred once in Eastern Province, part of the country, in 2015 after heavy rainfall. Transmission of this disease basically subcutaneous inoculation is the major transmission mode through direct contact with contaminated soil. So most cases are resulted from skin inoculation, and specific occupation groups like rice farmers, agricultural workers are more prone to the infection. Airborne infection transmission also possible. For example, during typhoons, cyclones, airborne transmission of the disease have been reported. Heavy rainfall, floods also facilitate the disease transmission. Ingestion is one of the important transmitted mode because the water can contaminated with this bacteria. So MLST is a widely used epidemiological tool which can characterize the genetic diversity of pathogen population. Here in P. pseudomallei population, we characterize using this technique. This technique is based on the variation in 7 housekeeping genes. There are several bioinformatic tools have been developed. For example, eBurst Phylogenetic analysis to elucidate the genetic relatedness of pathogenic organism using their MLST data. Here we used B. pseudomallei data from different locations in country and for endemic regions. The aim of the study was to determine the genetic diversity among Sri Lankan clinical isolates. Objectives of this study was to perform MLST of 87 clinical isolates of Burkholderia pseudomallei and to perform eBurst analysis for all Sri Lankan clinical isolates so far reported from Sri Lanka using MLST data. Ethics approval was obtained from Ethics Review Committee, Faculty of Medicine, University of Colombo. Materials and methods. Methodology is a bit complicated, therefore I made a flow chart. So we started with the work actually obtaining culture positive clinical isolates, culture isolates from 87 clinical isolates from University of Colombo. We extracted genomic DNA and we performed PCR amplification of 7 housekeeping genes using previously described oligonucleotide primers. We checked for amplification on 1.5% agarose gel. After confirming that we have amplification, we submitted all PCR products to the sequencing, which was done at Macrogen, Korea. So we obtained raw sequences. So we need to edit those sequences and we need to trim those sequences. So we performed trimming and editing of sequences and then we submitted all allele sequences to the pubMLST database. Database is providing the allele arbitrarily numbers to the 7 alleles. And then this is called allele profiles. Allele profiles, then we need to submit to the database to find the matching STs. STs means sequence types. So we submitted all allele profiles to the database and we found sequence type for most of the isolate, but we did not get ST types for 22 isolates because matching STs were not available. Therefore, we had to submit those data to the curator and the curator will analyze those data. Curator analyzed those data and provided us the novel STs. So now all ST data we used for our analysis using eBurst, Phylogenetic, and then sequence data, concatenated sequence data we analyzed using phylogenetic analysis in MEGA. So results, MLST analysis yielded 43 STs with 22 novel STs. eBurst analysis yielded 4 groups basically. Group 1 is having 46 STs distributed among subclones and subgroup founders. This is the largest group. Group 2 and Group 3, Group 4 are having few strains. In addition, we found 17 singleton, which is not having any relationship with the existing sequence types. So this is the result of eBurst analysis. As you can see, this is group 1. We call this as a clonal complex. In blue shaded areas, double locus variants are present. In red shaded area, single locus variants are present. The middle circle is the founding genotype. In Sri Lanka, founding genotype is ST1132 and is distantly related to the other subgroup founders. These are subgroup founders. So E-burst analysis resulted a considerable genetic diversity among 87 Sri Lankan clinical isolates of B. pseudomallei. This is a result of phylogenetic analysis. For this analysis, we used STs from other endemic regions such as Australia, Thailand, Malaysia, China, India, Cambodia, Vietnam, including Sri Lankan STs. We found there are two major clades, one for Oceania, another for Southeast Asia. Our strains mostly lies between Oceania and Southeast Asia, and we identified two distinct clades therefore. This may be, this could reflect the geographical isolation over a long period of time. This is the minimum evolutionary tree constructed using neighbor joining method in MEGA. For this, we used concatenated nucleotide sequences as input sequences. And we determined phylogenetic relationship of our Sri Lankan isolate along with the selected isolates from other endemic region based on the E-burst analysis. Here we can get more resolution than E-burst analysis because E-burst analysis is based on arbitrary numbers, but here minimum evolutionary tree is based on nucleotide sequence diversity. Therefore, we were able to obtain a phylogenetic relationship for those 17 singleton which came as singletons in MLB E-burst analysis. We found phylogenetic relatedness of STs from India, Bangladesh, and Cambodia. In conclusion, Burkholderia pseudomallei population in Sri Lanka appears to be intermediate between Southeast Asia and Oceania. Several shared STs we found with Australia, Thailand, Cambodia, India, and Belgium. And as observed in previous studies also, this could be due to homoplasy. So migration of humans, animals also contribute to the dissemination of shared STs among these geographical locations. Phylogenetic relatedness with STs from India, Bangladesh, Cambodia may be partly due to the multiple introduction of B. pseudomallei through anthropogenic sources, especially travel and trade routes. These are the references. We acknowledge the technical staff in the Biomedical Laboratory 1 at KDU and in the Department of Microbiology, University of Colombo for their valuable support. Thank you.
Thank you, Dr. Jayasinha. Your presentation now is open for discussion and suggestions.
I'll start again. I think the word homoplasy, I would like you to describe what that means.
Yeah, actually, so homoplasy is characters shared by set of species, basically, but not the common in their ancestor founding. So here, ST types are similar, but those strains are genetically dissimilar, diverse. Only the ST, only sharing. ST is based on actually, sir, 7 housekeeping genes. Housekeeping genes are getting very less variations between generations actually. Therefore, they are not genetically similar. Actually, they are highly diverse, but ST types, only one character, they are sharing. That's why we said that actually that term is differentially explained in different cases. In here, actually, in multilocus sequence typing, we described like this. Actually, those strains are genetically not similar, but STs are similar. ST are based on, it's a genotype based on, derived from housekeeping genes, actually. That's,
Thank you. Now, in the last paper, you said that after the second paper, you might make a comment more about the clinical correlates of these findings.
Yes, sir. Actually, yes, sir. We actually recently submitted one paper for clause neglected, so it was accepted. In that paper, we have used 310 population, because in my student Kumari's presentation, she used only 52. In our case, actually, sir, we have a neurological melodiosis cases. In Australia, basically 15 of the population is having
that bimA variant, it is coming from Burkholderia malai. It was a more virulent strain during the World War, actually, that was used as a bioweapon to make a glanders caused in horses, actually, during that time. So that is that strain cannot prevail in the environment, but in this Burkholderia pseudomallei can prevail the environment. So having B pseudomallei, bimA variant, malai variant in Burkholderia pseudomallei gives a more virulent form for this bacteria, since this bacteria can even prevalent in the environment. So anybody can use as a biothreat agent also, unlike malai. So everybody is interesting basically. So this is a purely a genotyping study. In genotyping study, basically we wanted to characterize, actually, we want to see genetic markers which could help to locate the Sri Lankan isolates within the broad geographical boundaries of B pseudomallei, because any outbreaks can be predicted in future. That was the main idea. So in our study, actually, with a large population, we clearly identified bimA is correlated with significantly correlated with the neurological melodiosis.
Thank you. That was very clear.
Yeah, thank you.
I raised a question in one of the earlier presentations.
Yes, madam.
Yeah, and I think, as you said, that the explanation might be here. Now, I think you're talking nearly more than 300 cases at the moment, isn't that right?
Yes, sir. Yes, madam. 310 cases we have used.
Yeah, over how many years is that? How long did it take to? I know that Colombo Medical Faculty, I think Professor Correa started, and also as explained by Dr. De Silva, Professor Tevanesan started some work. So over how many years is this? Do you have a time trend slide for this? I mean, how?
Yes, madam. This is, yeah, actually we are working with the collaboration of Dr. Inoka Correa. This strains actually reported from 2006 to 2018 strains we considered. Now our database, actually we have more than 600 strains. Out of that we used 310 strains because of financial constraints, actually, because MLST analysis needs huge amount of money for sequencing and genotyping study. Therefore, we have now, madam, within last 2 years, I think we found more than 200, 300 strains. Now the cases are reporting in Sri Lanka. Because
what is the total cumulative number of cases? I think that's what we are interested as clinicians. I listened to the oration by Inoka about a year and a half ago. What is the total cumulative number of cases in Sri Lanka?
Now, sir, around 600 numbers are there in our database, but all cases are actually not reported to the Colombo University.
This is because of the fact there is surveillance going on, is it?
Yes, and no, yes, madam. And one thing is, actually in Colombo University, there is, I think, latex agglutination test that laboratory is conducting. So more samples are coming to that. It's a main center. Surveillance is also going on. But whatever samples are not coming, will not be recorded there. So more cases must be there around the country.
Okay, because the word that was used earlier was that it's an endemic disease. So
yes, madam.
I don't know whether we are justified in saying endemic unless we know that there is a known number of cases roughly occurring every year within a time zone over a period of years to classify them as endemic. So that's why I said whether you have a slide on time series. I know this presentation has nothing to do with it, but that is out of interest, so that when you call a disease endemic, you have to be very careful in saying, oh, are they sporadic? But I think now they have gone beyond being sporadic. If you have got 600 and probably half of them identified within the last 1 or 2 years. Yeah?
Yes, madam, because Sri Lanka has now named as an endemic country to the melodiosis.
Yeah, that is what I wanted to clarify. Okay.
Yes, madam.
So,
Imali, I hope you don't mind. I think
yes,
the correct name of the disease is melioidosis.
Yes. Thank you.
Okay, thank you very much for your clarification.
Thank you. Thank you, sir.
And madam, if I can come in here,
yes.
Professor Navaratne, for your question, I think one big reason for now this melioidosis to come up in the recent is that because of the diagnosis. Earlier, probably these were probably misdiagnosed as something else, maybe like a pseudomonas, and treated and somehow without being noticed that it's a different strain. So now with the Prosinoka Korea's work and Tevanesam work, the lot more laboratories are aware of Burkholderia pseudomallei and their identification. So that's why we are saying it. So all these years it would have been there in the background, being unnoticed or not being reported because of the poor laboratory diagnosis, because the awareness and the resources were not there to identify that.
Okay. They would have been reported as sepsis or something like that.
Sepsis, and sometimes
in the oration she mentioned all the possible diagnoses under which it could have gone in the past without being recognized.
Yeah, it could have been easily diagnosed as a Pseudomonas species because it's an oxidase positive. So that's a simple test we can do in labs, and that will go on under that.
Thank you. That's very interesting and good work. Thank you.
If I can just add to that, madam. So when Dr. Inoka Korea was doing this, I mean, we didn't have the funding, and actually that's when I came in, and I mean, not to try to blow my horn, but because we were able to get the US funding, actually the US Army Institute, they actually took a chance because we hardly had any cases, because Professor Tevanesam had found 1 or 2 cases. And then I happened to be working with Dr. Inoka also. And they said there's hardly anything there, but then we knew it had to be around. I mean, Inoka was very convinced. I mean, I didn't know at that time. And then somehow they also took a chance. And then if you look at the numbers, I don't think Himali was able to show that slide. Around 2012, when the grant started, the cases skyrocketed. And all of that was able to get it. And it's very unfortunate that the government is making it so hard to get foreign funding now. And it has actually gone up and
Darshan,
down.
I hope we are not helping the Americans to develop a bio-weapon. Right? Because they might unleash it on us also one day.
Yeah. No, so you asked me that question earlier also. Actually, we don't send any of the strains there. And the main reason, even while I was at GeneTech, even though we were the main recipient of the grant, the PI, Inoka was also there, all the testing and everything was done at Colombo University because of the safety. And she keeps all the strains. Even today.
So no biomaterial goes to the States?
No. And actually, especially, they didn't want to because of the danger, because it is classified as a biosafety level 3 agent. They actually didn't want any of the strains coming there anyway. They happily gave the money. They want us to do the research and publish. That was the key thing. And we did that. And they were actually quite happy with it. Unfortunately, during my transition, we lost the grant. Otherwise, we would have had a lot more money to continue with this work, and we would have had a lot more data to share. So that's the background.
I hope, Darshan, that it's sorted out and you will get
yes, madam. Actually, even just now, an hour ago, I called the MOD to push them to put the cabinet paper. So we are working on it. Hopefully, we'll get it. Thank you.
Rohini madam, I just want to add something. Actually, this list we have made this database based on the culture positive cases. All are not culture positive cases. Some diagnosed with the agglutination positive, and they have symptoms and everything and treated for the melodiosis. But culturing is difficult for this bacteria. That's why we are getting less number. Only culture positive cases we are maintaining the database. Thank you.
Okay.
No, and just for their accuracy, in the 2 papers, the case fatality rate was given differently. In the first one, it was mentioned as 40, and you mentioned it as 24. I think 24 is probably the more accurate one.
No, sir. Yes, sir. I just
based on just 52 cases. I think it was 47%. This one had 24 with larger number, I think.
Yeah, because she has selected the 50 strains that depending on the study population, that figure. Actually, in the world, other endemic regions, it was 20 to 40 depending on the settings. In Sri Lanka, currently, it was around 24% because my students study actually very less population. We can't make any inferences.
She gave the case fatality for her case.
It's by accident, actually, her 52 contain more death cases. That's why. Thank you.
Right. That's very interesting.
Thank you, sir.
So what is the diagnosis? What is the treatment if you have diagnosis?
Yes, yes, a non-antibiotic course is there.
Actually, can I add to that?
Yeah, yes.
So actually, this is a very long course of antibiotics you need to give. It's
what do you use, Kilini? What is the agent you use?
Actually, that is depending on the sensitivity pattern. Mostly, it's ceftazidime plus a cotrimazole or doxycycline. So it's a combination of products. So most isolates are resistant to ceftazidime, and then you have to go for meropenem or imipenem. And there is an initial course and a continuation course. All goes up to something like 3 or 9 months sometimes. It's a prolonged course of antibiotics. So this organism is used to cause deep-seated abscesses.
Yeah.
Bloodstream infections and deep-seated abscesses anywhere in the body. So it's actually now, it organism is intrinsically resistant to aminoglycosides. That is this gentamicin, amikacin, etilmycin. So this group is intrinsically resistant, and also it's intrinsically resistant to colistin. So that's how in the lab we suspect.
So Dilini, cotrim is emerging again as a useful
yes, sir.
Agent. Is it in so many resistant organisms?
Yes, yes, cotrim as well as tetracyclines.
I see.
Thank you.
Any other questions? Any other questions? If there are no questions, we can conclude this session. All right, we'll conclude this session now. And I would like to thank all the presenters for their time and coming here and presenting their study findings. And we talked about fetal hemoglobin inducing agents in management of beta-thalassaemia. Then we had several discussions on SARS-CoV viruses, and especially the strain isolation in Sri Lanka in the cohort in 2020, and then another new variant which was found in Sri Lanka in a particular family group. And then we had in discussion on Burkholderia pseudomallei, which is of interest right now because of the increasing detection of this and their significance and their genetic variations in comparison to the Asian and Australian strains. So we had interesting discussion with contribution from the audience as well, very valuable contribution from the audience as well. So I would like to thank all the presenters again and the audience as well for contributing to this discussion in a very significant way. And the judges as well, Dr. Jayantha Balawardena and Professor Abhay for being with us and doing the judging of the presentations. So thank you all, and I will we'll leave for today now. Thank you.
Thank you very much.
Thank you.
Thank you.
Ladies and gentlemen, we have come to the end of the last technical session, which was indeed very interesting. On behalf of the organizing committee, I would like to thank Dr. Varuna Navaratne and Lieutenant Colonel Dr. Manoj Bandara for chairing this interactive session, and to the judges, Professor Vimaladharma Abhevikrama and Dr. Jayantha Balawardena. With this, we conclude the fourth and final technical session of the 14th International Research Conference of General Sir John Kothalavala Defence University.
Thank you.
Ladies and gentlemen, now we have come to the end of this grand event and hope that all of you have enjoyed the International Research Conference of General Sir John Kothalavala Defence University.
These 2 days have indeed been revitalizing and mind-stimulating, creating many opportunities to spark interest in many pertinent areas of conference was to capture these transformations from every aspect of academic and policy lenses, from disciplinary lenses of defense, legal, medical, engineering, technology, management to social sciences.
In the spirit of adapting to the new normal, this year, the International Research Conference was held for the first time ever virtually, with a total of 130 participants, and I am pleased to say that it was a success. Now, Air Commodore Professor Nama Vijay Singha, Professor of Medicine and Dean of the Faculty of Medicine, General Sir John Kothalavala Defence University, will deliver the vote of thanks.
Good evening to all of you. IRC 2021 of KDU is coming to an end after 2 successful days. And this is the 14th consecutive conference we had. And I was fortunate to take part in at least 11 of them as one of the earlier members of the Faculty of Medicine. The significance of the conference this year is that we were able to hold the conference fully virtually, though, in spite of all the difficulties we face in the ongoing pandemic all over the world. However, with all the efforts by many personnel, we were able to conduct this conference successfully during last 2 days. First of all, I thank the Vice Chancellor of KDU for his tremendous leadership and encouragement given to all of us to continue with these activities. In addition, the 2 Deputy Vice Chancellors, the Deputy Vice Chancellor Defense and Administration, and also the Deputy Vice Chancellor Academic. I thank them both for their support given to us. I must thank the conference chair, Dr. Harinder Vidhanage, for his leadership, for organizing this massive event, and also including the medical faculty as a part of it. My sincere thanks goes to a special person in the medical faculty who is none other than Dr. Prasad Premaratne, who is the ICU main, the medical faculty IRC main organizing committee of the medical faculty coordinator. Lieutenant Colonel Dr. Prasad Premaratne showed the true leadership and took us all to this success. His team, namely Dr. Amali Dalpadadu, Dr. Anuji Gamage, Dr. Sachini Rasnayaka, and Dr. Sachitra Ilangantilaka, all supported him well. In addition, I thank the chairperson of the abstract review and the publication committee, Dr. Priyamali Jayasekara, and also the chairperson of the editorial committee, Dr. Varuna Navaratne, and their teams for their excellent performance in selecting the research work to be presented. In organizing the events, I thank Dr. Darshan De Silva for organizing the plenary sessions, Dr. Varuna Navaratne for organizing the poster session, and also Professor Ayendra Balasuriya for organizing the technical sessions. And there are several other people involved in invitations and hall arrangements and registration desk and so on. I don't mention the names, but I thank all of them for their efficient work. I thank the chairpersons in both plenary sessions as well as technical sessions for their good work, as well as the judges of the technical sessions and the poster sessions for their contributions. This year we had a very successful plenary session where there were 4 eminent speakers. I thank Senior Professor Rohini Fernandopulle, Dr. Pali Tabekon, Dr. Chandima Jeevandara, and also Dr. Tushan De Silva for accepting our invitation and giving excellent presentations during the plenary sessions. In addition, I also thank all the presenters and the investigators for selecting KDU IRC to present their high quality research and submit abstracts. And also all the participants, both in person here as well as online, not only from Sri Lanka and also from other countries, for their participation and also contribution during the discussion sessions. I want to thank a special person. Without him, we wouldn't be able to conduct this successfully, because he looked after all the technical affairs. I thank Mr. Nilan Sanjeeva. Mr. Sanjeeva, I thank you so much, because last few days you worked tirelessly to make this event a success. And I also thank all the medical students, both officer cadets and also the day scholars for their contribution in organizing and also comparing the session. So with that, I thank all of you, and we will look forward to a successful, a more successful and a more glamorous KDU IRC next year. And also I request all of you to stay safe and continue to work hard for the betterment of our country. Now, as the last event, I think the winners of the poster session and the oral presentations are ready. We got the results just now. The winner of there were 22 posters presented at the medical faculty sessions, and the winner or the best poster was selected as abstract number 9, Evolution of Fatal Lung Damage in COVID-19. The authors were S.D. Arachi and R. Ruwanpura, and the poster was presented by R. Ruwanpura. Congratulations, either Mr. or Mrs. Ruwanpura or Dr. Ruwanpura. Dr. Ruwanpura. Okay. This is our own Rohan Ruwanpura. I can see him now. Yes, congratulations, Rohan. Your poster was
thank you.
Your poster was the best poster. And oral presentations, there were 24 oral presentations, and the best presentation is abstract number 627, and that is identification of new variant of SARS-CoV-2 virus of C36 lineage with L452R mutation at University Hospital KDU. The authors were H. Abegunawardena, UTN Senaratne, MMPT Jayasekara, WMID Nakavita, FN Mubarak, US Kulasekara, DGP Kavyangana, and AD De Silva. And the presenter was H. Abegunawardena, that is Mrs. Harsha Abegunawardena from the Clinical Department of Clinical Sciences, KDU. Is she there? Is Harshi connected? Looks like she isn't. Harshi is there. Harshi, you are the winner of the best oral presentation during the technical sessions, and I heard that few people were commenting that your presentation was crystal clear, and that was identification of a new variant, which is also a very important thing that you have found. So congratulations.
Thank you, sir.
So with that, I think we will conclude the KDU IRC 2021 to an end. Thank you very much, and stay safe.
Thank you.
Thank you.
Thank you, sir. I would like to express my appreciation to the plenary speakers, oral and scientific paper presenters, the poster presenters, and all of the participants for taking time off their busy schedules to attend the 14th International Research Conference of General Sir John Kothawala Defence University.
Our sincerest gratitude towards the Vice-Chancellor of General Sir John Kothawala Defence University, Deputy Vice-Chancellor Academics, and Deputy Vice-Chancellor Defence and Administration, Dean, Faculty of Medicine of General Sir John Kothawala Defence University, the main organizers of this event, Dr. Harindra Vidanage and his team, the faculty organizers, Lieutenant Colonel Dr. Prasad Premaratne, Dr. Anuji Gamage, Dr. Amali Dalpadadu, Dr. Sachini Malaviarachi, and Dr. Sachitra Illangan Tilaka, and to the several subcommittees involved in making this grand event a success. And my sincerest apologies if I had missed anyone out.
Finally, we have come to the end of the International Research Conference for the year 2021.
We hope each and every one of you enjoyed and broadened your knowledge as much as we did. Thank you for joining with us.